Welcome everybody to another really exciting edition here of our biochemistry journey. We're going to be learning today about uh, taking a step away. Over the last several weeks, we've been learning an awful lot about how we eat sugars and what we do with sugars. But we just had Thanksgiving, and so you guys probably have had a little bit of Thanksgiving dinner, a little bit of turkey, and you're probably thinking about, you know what? I'd really like to learn about amino acid metabolism. Where do all these proteins come from? And how do I turn them into yumminess? And why do I get tired when I eat tryptophan, right? No doubt this is what you're thinking, the burning questions they were thinking about over Thanksgiving. I know how that goes. Let's learn about amino acid catabolism and anabolism. Today, we're going to first learn about how we make amino acids, and then we'll talk about how we break amino acids down. Okay, so today we're going to work on the making of amino acids portion of that. That's amino acid anabolism. Anabolism is to make, catabolism is to break down. And so without further ado, let's get into it. So as you guys have probably heard before, and you're looking at your nutritional information on the side of your cereal boxes, that there are these things that they call essential and non-essential amino acids. You've probably heard this before. Yeah, have people seen this before? You heard of essential and non-essential amino acids? Yes, yes. You ever wonder what the heck that means? Like, what does it mean to be an essential amino acid or not? Like, it means in biochemistry terms, like whether you can make it from scratch or not, or whether you have to rely on it from your diet, right? And nutritionally, that seems to make a lot of that's a fairly important distinction. You either need to eat it or you can make it from scratch, right? And so the essential amino acids are things that we as humans don't have the biosynthetic capacity to make anymore. We've lost it through evolution, and then the non-essential amino acids are things that we can make, and we still have all the full biosynthetic capability. Now, there's a third category here, which is kind of squishy category called conditionally essential. So what that means is that that you have the biosynthetic pathways to make an amino acid from a precursor, some specific precursor, but maybe not from scratch. So, and we'll talk about a couple of examples of those. But if you only need to modify a substrate a little bit to make another amino acid, then that would be, but you're dependent on the original substrate, then that would be conditionally essential. Okay. So why have we lost this biosynthetic pathway? What do you guys think? Why is it that humans don't have all biosynthetic pathways in us at all times? Chat, what do you think? Why don't we maintain our 100% of our metabolism? Why are humans dependent on?
Not enough energy to make all of them.
Well, yeah, I mean it does require energy. Daria is interesting because we can just eat it. Our diet supplements some of the amino acids, as mentioned by Andrew. There's no reason to have it, so why waste resources? So I think you guys kind of have the gist of it, which is that we eat whole organisms, right? We eat plants or we eat animals. And when we do, we more or less get all the amino acids in a go. So there's not a whole lot of reason evolutionarily to maintain all the biosynthetic pathways, because we're not eating things like ammonium and synthesizing carbon from carbon fixation, right? We're not a plant. We have the ability to go out and eat plants. So, we don't really need those biosynthetic pathways. So that's really what's at stake here. And so not surprisingly then, the pathways that are the most complicated and have many, many, many steps in biosynthesis tend to be the ones that we've lost in our evolution. Yeah. So here's a quick look at the non-essential and essential amino acids. So these non-essential amino acids here are, alanine, asparagine, aspartate, glutamate, serine, which we're going to see how each of these are made from biosynthetic intermediates. These conditionally non-essential ones, this category means that we're requiring or relying on a particular substrate to make them from arginine, cysteine, glutamate, glycine, proline, tyrosine, right? And then you have these things that we're totally dependent on the environment to, our diet to have with us. And actually, we had quite a few in the human diet. As we'll see, that this is not something that applies to microbes or plants. So when we go and we look at plants or microbes, they generally can make all of them, right? So this is, we're going to talk a little bit about what humans do and a little bit about what plants do here in this lecture. Okay, so something that's different between humans and free-living bugs is that by and large, free-living bacteria have to contend with whatever they have available. And so they tend to have more complete biosynthetic pathways than we have. So when they become dependent on a particular amino acid, it's unusual, and we call them auxotrophs, okay? And so an auxotroph is something that is dependent on an exogenous source of that amino acid to grow. For us, we would call this being a human being, and these are essential nutrients. But when you're talking about Staph aureus or something like that, and it requires exogenous arginine, we call it an auxotroph, and we consider it a sort of a genetic mutation or genetic, deficiency, right? So these experiments are typically done by, you will grow, you can illustrate auxotrophy by, you might have a minimal media where nothing will grow, and a prototroph, which means something that has all of its biosynthetic pathway, can grow on that minimal media, but an auxotroph cannot, right? So if you add back a specific substrate like arginine, you can then find all of those colonies that are allowed to grow on that. And that's just demonstrating that this is, in fact, an arginine auxotroph mutant. And this is shown here in our control experiment where we add lysine and nothing grows. Anyway, the point here is that this is a way of demonstrating which kinds of amino acids are essential for particular bugs. This is kind of an interesting phenomenon. This is a fairly important kind of phenomenon, particularly when it comes to clinical things. So 30% of the isolates that come from people's lungs with cystic fibrosis, the microbes living in their lungs, have auxotrophies in their biosynthetic pathways, something like 30 or 40%. And many of them are in the very same biosynthetic reactions. So there's a very good chance that there's some sort of intersection between the evolution of these auxotrophies and the emergence of career pathogen microbe strains. And so this is kind of the Wild West, one of the things that we study in our lab, but is really pretty interesting. The thing is, this is what some real world data would look like in discovering an auxotroph. Here we have a bacterium that's a histidine auxotroph, and we can show which step along the way the bacteria has become resistant to or has become dependent on, because we can just supplement in histidinol here, this last biosynthetic precursor, and show that you can recover the growth of these bacteria with just this medium. So, okay, so as an overview, the amino acids are by and large synthesized from things that you guys have already seen. The reason why we covered the TCA cycle and recovered glycolysis and have covered these things to make sure that you know them is that you're going to see that most amino acids are made from your friends, your biochemical friends. What do you mean by career? Robin, I was just talking about opportunistic pathogens in that comment. So all the amino acids ultimately get made from things in central carbon metabolism that you guys already know about. So there are a handful of standard entry points that we'll see result in some different categories of amino acids. So alpha-ketoglutarate is this 5-carbon precursor, and it ultimately makes this sort of family of amino acids that comes out of it. Pyruvate, as you remember, is a 3-carbon precursor, and that ultimately makes alanine, right? Or we have this 4-carbon oxaloacetate, and that makes this 4-carbon aspartate and asparagine, like these guys here, right? And then we have some other really interesting pathways. These are microbial pathways that we're going to learn about here that ultimately come from charismate. So charismate making these really interesting aromatic amino acids of phenylalanine, tryptophan, tyrosine. We'll learn about that. And then this other 3-carbon sort of route that we have for making serine, glycine, cysteine. So basically, we have we have these branch chain amino acids. We've got this sort of family of things that is derived from alpha ketoglutarate. We have a family of things that's derived from oxaloacetate. We have a family of things that's derived from charismate, and we have a family of things that's derived from 3-phosphoglycerate. And that's how you make all the amino acids. That's pretty cool, right? You have all these 20 amino acids that look so structurally diverse, and yet we just make them from the things that you've already seen before. Yeah, that's pretty clever in biochemistry to have been able to do that. You guys stoked to see how that works? Stoke. How's it work? Please tell me. I'm sure that's going on. Everybody rushing to the keyboard. Please tell me. How does it work? Yes, we have much excitement. Well, one of the things that you got to learn about is NADPH. Now, we've encountered this once already, NADPH. What does NADPH do? Reminder from the chat, what does the NADPH do? It was from ancient history, like 3 weeks ago, we talked about this. Yes, Jeremiah, Robin, yes, yeah, it's used in the pentose phosphate pathway for providing reducing equivalents. Somebody already had it right up there, though, that Haley's right, that this is also, NADPH is largely used for biosynthetic reactions in the cytosol, right? So it's a little different it's structurally nearly the same as NAD, except for this one additional phosphate, but it's frequently used as the source or the sink for electrons involved in biosynthetic reactions. Why do we have it, and why is it different from NADH? Well, obviously, this allows us to regulate biosynthetic reactions independently from catabolic reactions. Now, does that make sense? Separate energy metabolism from making things. Yeah? Okay. So before you can start, like, what the heck is an amino acid, right? What is an amino acid, chat? What makes something an amino acid? Huh? Thoughts? I hear crickets in the chat. Chiral center. What makes something an amino acid? Let's take a guess. What would it have to have in it to be an amino acid? We have lots of quiet. It has an amine. Yes. Ha 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 ha. And an acid. Yes, chat, you've got it. That's exactly right. That's all that's required for it, is to have these two functional groups. It has this, a carboxylic acid, which is typically a minus charge at one point, and it has an amine, which is typically positive under the conditions that we look at. So this is all that's required for it to be an amino acid. And so they have this general structure. Whoops, go back. They have this general structure. NH3 and then R, right? So they have this general structure that we have. We put there are 20 different side chains that we put on that R group, right? As you recall. But if the defining features are carboxylic acid, where does the carboxylic acid come from? Chat? Where does the carbon chain come from? Chat. Hello. Where does the carboxylic acid come from? Does it come from things that we've recognized before? It comes from these organic acids that we've already seen in glycolysis and TCA cycle, central carbon metabolism. Yeah, that's exactly right. Jeremiah's right on it. This is a central carbon metabolism it comes from. And so if you're a microbe, then and you want to make an amino acid, then what do you have to find? You've got to find have to find an amine to put on there, right? And so how do you get the amine? So the very first step in amino acid metabolism is just how do you get inorganic nitrogen into your body and integrate it into a form that can be used. And so the way it comes in is it comes in from in microbes comes in as ammonium. Like this is not how we acquire nitrogen as humans, but this is how nitrogen ultimately becomes organic nitrogen as one of the, ultimately gets to you as required this reaction. So, so this reaction takes a glutamate dehydrogenase reaction. It takes a alpha ketoglutarate, this 2-oxoglutarate, that's another way of saying alpha ketoglutarate. Glutarate, okay, and ultimately takes this with an NADPH, makes a glutamate. So it takes a 5-carbon skeleton and a ammonium ion, inorganic ammonium ion, and then sticks it onto a 5-carbon unit, makes a 5-carbon amino acid. Yeah, whee, that's pretty cool. There's another route which has this, is called the glutamate synthase reaction. Glutamate synthase reaction, or GOGAT, is a terrible mouthful, which is another route of interconverting these. So we can take a glutamate, okay, and with a glutamine synthase reaction and an ATP synthetase reaction. Tase. For those of you who don't know, a synthetase reaction, that tase means that it's hydrolyzing an ATP. That's what the name means. And so it'll take those two things and it'll make your glutamine. Yay! You have a glutamine from a glutamate. Then once you have a glutamine molecule, you can take a glutamine and an alpha ketoglutarate, which, as you recall here, the alpha ketoglutarate is also 5-carbon, right? This is 5-carbon, and with an NADPH, you can then interconvert these to make 2 glutamate molecules, right? So one glutamine becomes 2 glutamates. Makes sense. There's a side chain glutamine, and there's a backbone, there's a side chain ammonium, and there's a backbone ammonium. And if you take those 2 ammoniums and you distribute them onto 2 carbon chains, 2 5-carbon chains, you can end up with 2 glutamate molecules, each of which has 1 ammonium, right? So this is the basic routes for getting ammonium into your body, as you have this glutamate dehydrogenase reaction and then this GOGAT reaction, and that helps move nitrogen from an inorganic form into an organic form. Yeah. So once you've gotten to glutamine or the glutamate, you can get to an awful lot of other amino acids, right? So here's our friend alpha ketoglutarate we were just talking about a second ago, right? And so we had our glutamate dehydrogenase reaction that got us here to glutamate. And then from there, the neat thing is that, we just talked about glutamine, but we can also go from there off to arginine proline, aspartate, and alanine. So we'll talk about how we go, particularly in these 2 reactions here, these transamination reactions, to make alanine and aspartate next, okay? So these are part of this exciting transaminase family. So transaminases are a cool set of enzymes. They're things that actually we look for if you guys have ever heard of if you've ever had your blood levels checked, you like get a blood workup done by the doctor, they'll look for aspartate aminotransferase. That'll be one of the things on there. The reasons for that is when you have a heart attack, their heart cells spill open, and one of the things that they find, the enzymes there, is aspartate aminotransferase or alanine aminotransferase, which isn't supposed to be running around your blood, but when you have a cardiac infarction, then your cells die, and then the interior contents go running around in your body. And so that's one of the ways they detect heart attacks. So what do these reactions do? Well, they take a alpha keto acid and then they interconvert it to they take an alpha keto acid and an amino acid, and they take the amine off the amino acid, and then they stick it onto another alpha keto acid. So you have this, nitrogen coming from here and going on to here at the same time, taking, interconverting these alpha keto acids and amine sources. And they all use this cofactor PLP, pyridoxal phosphate. And we'll talk about that here in a second. One of the things that's cool about transamination reactions is they have this ping pong mechanism where they have this two-step reaction where the substrates that are being used in the reactions will come on and come off, right, and then be replaced, come on and come off by a cofactor. So we'll show you a little bit about this. So this is the first example. This is aspartate aminotransferase. Now, so I just need to put a note here. You don't need to know mechanism here. And so I don't expect you to know this reaction mechanism. Other than that, what's cool is you have an aspartate coming in, woohoo, and ultimately you have an oxaloacetate coming out, right? And the way this this first step of this reaction, or this reaction proceeds, is through this little functional group right here. What is that thing called? Do you guys remember that? What's that thing called? You guys remember your shift base? Yay! Shift base, right? So this is just to prove that these reactions that you're learning are present all throughout all of biochemistry. So ultimately, the net effect of this reaction, though, is, and what you do need to know, is that the aspartate here will take its nitrogen, ultimately transfer it onto an alpha ketoglutarate, right? And in so doing, make a glutamate molecule, right? And when it does, the aspartate carbon chain ultimately becomes an oxaloacetate, right? So alanine or aspartate aminotransferase takes an aspartate and alpha ketoglutarate and transforms it into an oxaloacetate and a glutamate, right? And it does that through this ping pong mechanism using this PLP as a cofactor. Does that much make sense? Chat? Everybody understand? Yes. Cool. Well, the cool thing about this is that's not the only reaction that proceeds via this mechanism. The other one is alanine aminotransferase, right? And so alanine aminotransferase will take a alanine and an alpha ketoglutarate, and then it'll make a pyruvate and a glutamate. Same thing here. This ammonium from alanine ultimately becomes the ammonium from glutamate, right? Or vice versa. These are freely reversible reactions. So when you when you add alt and PL, or when you add these substrates, then they'll freely interconvert. So what's the summary here? The summary that I'm trying to articulate is that you have alpha ketoglutarate and glutamate on two sides, and then you have alanine and pyruvate, or oxaloacetate and aspartate. And that's interconversions done by AST, aspartate aminotransferase, or ALT. Isn't that cool? So one little mechanism there, and of course these all require PLP, one little mechanism there allows you to get from glutamate, alanine, aspartate, or all their conjugate amino, their conjugate acids. So that's pretty cool, huh? Not so challenging there. Stoke is high. Enthusiasm is big. Yes. Okay. So it turns out that's not the end of it, though. So the branched chain amino acids have their own transferase, and they also do the same kind of chemistry. So glutamate is used in all these cases as a cofactor again. Then it just changes what the, well, darn it, go back. You have, you just have 3 different alpha keto acids that get used: alpha keto beta methylvalerate, alpha keto isovalerate, and alpha keto isocaprorate that ultimately become isoleucine, valine leucine, right? And again, these are just done through transaminase reactions. So pretty cool. If you got the biosynthetic precursor and you got a transaminase reaction, you can make, now what are we up to? 1, 2, 3, 4, 5 plus 5 plus glutamine and glutamate, 6, 7. We're almost halfway through the amino acid biosynthetic pathways, yeah? Is Stoke extremely high? Stoked, good. We have at least 1 Stoke person. Oh, good, 2 Stoke people, that's good. All right, well, let's not let the Stoke end here because the excitement goes on. As you can probably imagine, glutamine is has other things that it can go into. Sadly, as we progress beyond these couple of reactions I've told you about, things start to get a little more complicated. So how do you make proline? Proline, as you recall, is this funky cyclized amino acid. It's what we use it for in biochemistry, is to make tight turns and protein conformers, right? So how the heck do you make that? How do you make proline? Well, it goes through this series of reactions, involves 2 NADPHs, right? 2 NADPHs going in, and it goes through these funky intermediates, glutamate, semialdehyde, and delta-5 or delta-1 pyryl 5 carboxylate. So what do you need to know from proline biosynthesis? You need to know that it comes from glutamate. You need to know this. You need to know that it consumes NADH or NADPH. And let's see. Yeah, that's it. And that results in proline. So yeah, I want you to know that. I want you to know this, and I want you to know this. So three facts, right? Facts to know. Everybody clear on that? Precursor, product, number of cofactors it goes through. Yeah. Asparagine biosynthesis. Asparagine is kind of fun. So have you ever wondered why the intermediates aren't important? They are important, but we're going to see quite a few pathways here coming up. And so if you would like to learn them and memorize them, then I commend you, although I'll have to admit that I myself look them up when I need to see them. Being familiar with and having seen the names is good. And we may quiz you on those because you can look them up easily as you work through your quizzes. But ultimately, long term, what's most important to know about the amino acid metabolism is where the carbon comes from and like how hard it is to make. Does that make sense? Yeah. So let's talk about asparagine biosynthesis. So does everybody, like, what does this name remind you of, asparagine? Like, what does that sound like? Stinky pea. Asparagus. Yeah. Do you ever wonder why it has that name? So it turns out it's not a coincidence. asparagus or asparagine was the very first amino acid ever to be discovered. I believe it was 1801. It was discovered by a French chemist, and asparagus juice. And as it turns out, if you boil asparagus juice and then let it cool, particularly white asparagus juice, which is where it came from, there is so much asparagine in the solution that it'll crystallize out and fall out of solution. So it was the very first amino acid to be discovered. Its structure wasn't fully validated, I think, until the mid 1800s, 1860 or so. But like the structural formula wasn't sorted out. But yeah, pretty cool. First amino acid discovered, asparagine. How do you make it? So it's got a little bit of a difference. So it also has this set of chemistry where we start with an organic acid here, starting with oxaloacetate and making aspartate via that aspartate aminotransferase reaction that we just learned. But then the next step is that it has to put on a side chain amido group, right? And so this goes through an amido transferase reaction. This requires some activation. So it goes through an AMP activated intermediate. So we'll take a look at this here in a minute. But ultimately, it also gets its amine from glutamine. So glutamine reactions we haven't stepped away from just yet. We're still seeing things where nitrogen comes from glutamine, but it has to have this activation state in between. So that's important to know. How does this work? So one of the things that happens is that the asparagine synthetase reaction happens in this enzyme that's got these sort of two separate compartments. One is where you have this nucleophilic attack of the glutamine to release the ammonium. Well, it doesn't really want the ammonium flying around because you want to use it for later biochemistry, and you would prefer it not to run around. It's also toxic. So there's this ammonium channel that allows it to go to another side where it attacks a activated intermediate. In this case, it's an AMP aspartate intermediate. And then you can undergo this nucleophilic substitution reaction to additional elimination here to ultimately get your asparagine. Yeah, it's pretty cool. Couple part reaction mechanism. Okay, so what have we done? Let's do a little review. We've talked about how glutamine is made, how glutamine is converted into glutamate, proline, how that gets into ultimately we've talked about in previous lectures how that can be used to fuel the TCA cycle. We've talked about how aspartate can become, or oxaloacetate can become aspartate and asparagine. We've talked about how to make alanine. And we've talked about how we get the branch chain amino acids there have their transaminase reactions. So what we have left here, well, let's talk a little bit about this 3-phosphoglycerate becoming serine and cysteine and glycine. These are reactions that humans can do. So the serine biosynthetic pathway starts here with 3-phosphoglycerate, and then we go through this funky intermediate here, 3-phosphohydroxypyruvate. And then, not surprisingly here, we have a glutamate providing a nitrogen source. We have a PLP cofactor that is providing the nitrogen. And we end up with this 3-phosphoserine intermediate. And after hydrolysis of the phosphate, you end up with serine. So pretty cool. You start with 3-phosphoglycerate, you end up with serine. Okay, what do I want you to know here? I want you to know the starting point, 3-phosphoglycerate. I want you to know the cofactors, and I want you to know the product, cofactors plus source of the nitrogen. Yeah, does that seem fair, guys? Those are the things I want you to know. Naturally, if you want to learn the structures for 3-hydroxypyruvate, I am not discouraging you of that. And it may show up on a quiz since it's easy to look up. But long term, these are the things that are important to know: is how do you make serine? You make it from glycolytic intermediate. When you make it, requires energy, it's making this NADH in the process. Okay. So glycine, as it turns out, is just one more step beyond serine. And it's a case where we get to see this the appearance of a new intermediate here, which is tetrahydrofolate. So you've probably heard about folic acid, right? And as it turns out, folic acid and its regeneration is a pathway that's really important, and all kinds of things like folate deficiencies have a big impact on our immune function, and are also very upregulated in cancer metabolism. But so what does folate do? Folate catalyzes one carbon transfers. It's what it does. It serves as it allows these one carbon transfers to occur. And so ultimately here, it serves that same function here in serine, where you start off with this serine with that side chain, and you end up with this glycine without it, going through this tetrahydrofolate cofactor. Yeah. Then there's a whole series of pathways that are used to regenerate the tetrahydrofolate, and those ultimately produce an awful lot of NADPH, which is something that we mentioned before in one of our previous examples. Hey, come on. There we go. So then we have, so we've covered these guys, we've covered these guys, we've covered these guys, at least the upper portions of them. We've covered these guys, and we've covered that guy. Okay, what else do we have? Well, we need to talk a little bit about these guys up here. So these are guys where we start getting into trouble here with respect to our biochemistry. So we can make tyrosine, and we make it via this monooxygenase, which is a fancy way of saying a redox enzyme that uses a molecular oxygen. And so we take a phenylalanine here, and we can make a tyrosine. Not surprisingly, right, that in order to take a phenylalanine into to tyrosine, we have to oxidize in that position. Everybody makes sense? Yeah. Not surprisingly, also, then this involves this cofactor that has to be regenerated here, and this is ultimately the reducing power from this is coming from NADH. So phenylketonuria is one of the things that we're going to be talking about later. I'll actually come back to this on phenylalanine hydroxylase. But the inability to perform this reaction results in this PKU metabolic deficiency that some people are born with. And have you ever wondered why, they have these packets of sugar that says warning to phenylketonuria, like contains phenylalanine? The reason is that people who are missing this enzyme can result in dangerous accumulations of phenylalanine as a result of this deficiency. Okay, cysteine biosynthesis, and we'll talk about a few of these others that are out here on the more complex edge. So these guys, cysteine is made from methionine, right? So this is another case where we're dependent on having the precursor in order to make the product. If you have the precursor, then you can go through SAM, acetylmethionine. This is a very important molecule. It's used in a lot of methyl transfer reactions. You see this got this methyl group that sits here on the sulfur. It's used in a lot of methylation kinds of biochemistry. And ultimately, this makes this S-homosyl cysteine, and after a long series of reactions, gets down here to cysteine. You won't be surprised to see, this series of reactions here is you know, it starts to get more and more complex as we get here into the secondary metabolism, these complicated structures. So getting these sulfurs on and off is no trivial feat. What do I want you to know here? I want you to know the precursor and the product and the cofactors. Okay, those are the important things to know. You should also know about S-adenosylmethionine. It's an important molecule. Sam is a friend of mine. Okay, so that's sort of a very quick overview of what humans do. So plants and bacteria have the problem that they can't get up and move around, and they don't get to eat, you know, whole other organisms typically. So they have to maintain all of their biochemistry from scratch, de novo biosynthetic pathways. So this leads to some pretty wild chemistry. Yeah, before I get into this, like, how do you make these guys up here from scratch? So we talked about, we make them from like one step away, basically, in the case of, you know, in tyrosine. Now, how do you make them from scratch? Well, the microbes do that with this precursor called charismate. This comes from the shikimate pathway. This is a very famous secondary metabolism pathway, very important in microbes. Really interesting. There's actually an expert on this pathway here, Alexei Sevchenko, who does the biochemistry on this on the med school campus. Anyway, so charismate, which is this really critical intermediate, it's ultimately made from phosphonyl pyruvate and erythros 4-phosphate, and it undergoes this 4 reactions. It's actually done by a you know, polycistronic enzyme. It's all coupled together in some organisms. Does all these series of reactions just with one polycistronic enzyme. So it makes shikimate, which is the sort of key intermediate, and via 2 other steps of biosynthesis becomes this charismate, which is the ultimate precursor for all these amino acids. Charismate comes in, is ultimately turned into phenylalanine, is turned into tyrosine, you know, each of these reactions here requires the microbes to have, you know, special enzymes. Just like in the other pathways, I expect you to know, like, these other complex pathways. And I expect you to know the precursor, the products, and any cofactors that are required for making them. Tryptophan biosynthesis is even wilder. So, you know, it starts with charismate, and then it goes to this compound called anthralenate. We actually see this a fair amount when we do our biochemistry analyses of microbes. One of the things we look for is these intermediates of these pathways. So we see anthralenate fairly frequently as a catabolite of microbial metabolism, or something that's a marker of that. And then so it goes through this series of wild reactions, ultimately making an indole ring, which is should look familiar. And that indole ring is ultimately then added on to a serine to make this this final tryptophan, and then tryptophan synthase reaction. Same thing as before. You should know the precursor, the product, and any cofactors that are involved in the overall pathway. Lysine, methionine, tryptophan biosynthesis. They are these are things that are sort of more connected to core biochemistry, but go through reactions that we don't have. Again, they're complicated, so we've dropped them in our own biochemistry, but microbes don't have that luxury, and so they also go through these series of reactions. Same thing, you should know the precursor and the product, precursor and product, and any key intermediates. Yeah. And then the final one is branched chain amino acid biosynthesis. So this is recently become in vogue for supplements for people who like to weight lift, branched chain amino acids. Well, where do they come from? Well, they come from these reactions here. Ultimately, they begin with threonine, and they go through these threonine or these other organic acid precursors, and they're also ultimately become the building blocks of isoleucine, leucine, and valine. Yeah. So same thing. You should know where the carbon chain comes from and where the amino acids end up. So learning objectives. I think the most important thing to take home from this is that ultimately we harness inorganic nitrogen, and we as organisms take in organic nitrogen and we fix it into organic form, typically via the glutamine and through these reactions that form glutamine. And then that carbon or nitrogen gets out through all of biochemistry through these series of reactions that we've just covered today to make all 20 of the amino acids. In microbes, they have to make all 20. In humans, we cheat because we eat them. Now, in the next exciting installment, we're going to talk about how we break down those amino acids, and then we'll talk about why we might want to consider supplements or using various amino acids or things in our diet here. We'll look at the energy drinks and supplement market and nutritional supplements, all that. So that's on tap for next time. That's it for today.